Today we're going to run through the evidence base for H3.1. It's a tremendously exciting opportunity. It's going to be a whistle-stop tour, essentially through 6.5 years worth of data. Really what we're trying to do by pulling all this information together is link our work from the laboratory bench right to the bedside and set the stage for our future studies that we're incredibly excited to share with you and incredibly excited to see what those results will bring. Really, this is a process of bringing scientific expertise into the clinical arena to allow clinicians to identify patients who respond to therapies, to risk stratify, and ultimately to improve patient care and save lives. A very busy slide that we click through here. And what we've done is cohort our work into scientific work, clinical work, particularly in sepsis, but also highlight where we're picking up studies in preeclampsia, stroke and thrombosis, and then nod towards our future studies coming as well. We've started with our basic science, the models and the assays, and these are covered in detail in our review article, The Net Effect, published in Critical Care Medicine. And this really picks up where nets, neutrophil extracellular traps, are central in the evolving concept of immunothrombosis. And this is the key connection linking our innate immune system and our hemostatic system, and how these two previously independent pathways link together. And our work validates H3.1, shows that it's an excellent marker of netosis or neutrophil extracellular traps, and this work is providing the underpinnings for our regulatory approval. Neutrophil extracellular traps were first discovered in 2004. So in scientific terms, we're really moving quite quickly to bring this to the bedside. We published our key paper last year, Vargnies et al., from our laboratory in Belgium. This showed we have developed an analytical, validated chemiluminescent immunoassay for detecting neutrophil extracellular traps. This figure from the paper shows the immunofluorescence saying that H3.1 is picking up nets. The test does what it says on the tin. It is an excellent marker for neutrophil extracellular traps. It's fully automated, reliable, and can be processed rapidly to facilitate clinical decision making. The second part of that paper also helped us establish a normal range. We've continued to consolidate that work for our regulatory approval, but that's being picked up here in this slide. In patients who are sick with sepsis, we see a particularly elevated signal. The other work through our innovation laboratory in Carlsbad, California, has really been delving into the basic biology, understanding how nets unravel, and the patterns of DNA condensation. These are complicated studies, but they're really linking the science to the bedside. We're incredibly proud of our most recent paper, published by Kayford et al. In Frontiers of Immunology in June 2026. This paper is the culmination of a series of work showing that we can reliably activate neutrophils, we can reliably stimulate them, we can identify the patterns of chromatin condensation. Most importantly, that work is now using natural biological triggers which act as an excellent model or mimic for sepsis. This enables us to test new molecules and facilitate precision medicine and work with the pharmacological industry. The key part of the most recent paper is the link to the VANISH study and subspace studies. These are extremely well recognised, published, well-reviewed studies in sepsis. We have shown a link with genes activated in sepsis through the VANISH study, and in the subspace study, which is looking at genes related to the severity of sepsis, we've shown crossover there too. That's amazing, because it's linking the basic biology of neutrophil extracellular tract activation, H3.1, to the clinical severity of patients at the bedside. That's an incredibly strong biological model, and it explains why we can use this or hope to exploit this in the clinical arena to identify novel treatments and enrich the prognosis and understanding of patients. Volition's key goal has always been to bring our science to the bedside, and we've done that through a series of clinical studies in sepsis. That work has continued seamlessly in parallel with our scientific work. You can see here that we have 7 studies in the sepsis arena. We start off with our first papers published right at the peak of the wave of COVID. These were the initial signal showing that there was an H3.1 signal relating to severity of illness. That work has been taken forward with two very large studies. The first published with our colleagues at UMC Amsterdam in the Mars study group, looking at 1,700 patients with sepsis, showing a clear link between H3.1 and the severity of illness, with the presence of sepsis, and in particular picking up the hyperinflammatory subtype of sepsis. This is a tremendously detailed paper and shows the biological relevance and shows how we can use H3.1 to help identify the sickest patients. Our next large cohort is just under 1,000 patients studied with the SEPNet group in Germany, looking at 1,000 patients or 971 patients in 33 German intensive care units. Most importantly, the CIS-PCT results reinforce the results from our study in Amsterdam. We have a distinct geographically dislocated population, but the results are similar. There is a clear link with severity of illness. There's an increased hazard ratio and risk of death, and a clear risk and increased risk of renal failure. The CE marking for H3.1 is broad, as it's picking up netosis. There is a very strong netosis signal or disordered netosis signal in sepsis, but we can pick up that signal in many other diseases. And therefore we think the market and potential for H3.1 is huge. You see that signal because it is linked to the biology of diseases and linked to the principle of immunothrombosis we mentioned earlier. Therefore, we've seen a signal in patients with preeclampsia. We've seen a signal in patients who suffered strokes. And we're seeing an increasing signal in patients with trauma. And we're very much looking forward to picking up a second study with trauma later this year. Weaving together these two parallel streams of our strong scientific data and clinical data has enabled us to move forward with new studies. We are eagerly awaiting the results of the RSU record study with Professor Djali Anand, and that's looking at the use of H3.1 and corticosteroids in patients with sepsis. H3.1 gives us a new window into the biology of sepsis. This is helping us to identify the sickest patients. It's helping us to identify a treatable trait and should lead to increased precision and refinement of the therapies that we can give. The second study that we're doing with Professor Anand is the DETECT-SEP study, which should open in the autumn of 2026. This is using H3.1 for the first time prospectively in the clinical arena in French emergency departments to triage patients, to identify those most at risk of deteriorating and escalate their therapy appropriately, and the opposite, to identify those patients who do not need to be admitted and support their discharge home. This huge body of work, produced in just over 6 years, bringing together the biology and the clinical signal, is incredibly powerful. H3.1 is a treatable trait which will enable us to improve patient care and hopefully save lives. We're incredibly pleased to be working with Professor Djali Anand, one of the world's leading experts in sepsis, with many publications extremely highly cited and a huge impact on national and international guidelines over the last 20-25 years. I'll hand you over for a last few words from Djali.
It's the first time I have seen so many data coming so fast and with consistent findings. So what we are likely to see now with H3.1 and more globally with NETs measurements is a high likelihood of getting a treatable threat, a game changer in effectively modifying patients' trajectory.